All right, students, so this is the transcription lesson that I promised to record for you guys. Hopefully you can see everything clearly enough that it makes sense to you. I do apologize for a little bit of glare that's going to happen over here. I am going to be referencing my notes just as I did in class to make sure that I don't leave anything off. There are a few things up here that are labeled for you. We're going to go over what each of these things represent. I want to make sure I didn't leave anything off. Looks like I did. So let me go ahead and finish setting this up so that it looks just like it did when you were in class. Or if you weren't in class, then obviously make sure that it looks like what everybody else was able to see. Looks like we're good. All right, so what you're going to notice here is that we are looking at a diagram of a very simplified nucleus. All right, so we're going to label this as the nucleus so that we don't forget what we're looking at here. Obviously inside the nucleus, we're going to have DNA. Here's our DNA strand. There are several markers on here. I've also identified a section of it as muscle and a section of it as bone. I'll get to what that means here in just a second. But if you're setting this up in your journal, then if this represents the nucleus, everything else outside of the nucleus is going to represent the cytoplasm. So we're going to identify this area, if you will, as the cytoplasm. Now, there are a few reasons why DNA has to be inside the nucleus. We've talked about this before when we looked at organelles. We know that the nucleus contains genetic information, but hopefully you also included the idea or the fact that the nucleus is also protecting the DNA by containing it within the nucleus. So it protects it because in the cytoplasm we have an enzyme known as DNase. And as you recall, hopefully as you recall, anything that ends in ASE is an enzyme. Well, for the most part. There are a few exceptions to the rule, but this is an enzyme, and being DNase or DNase, it's an enzyme that is going to break down DNA. Ideally, we don't want to break down DNA since it contains all the instructions that we need for everything our bodies need to do to make sure that our cells are functioning properly. We're going to keep it safe in the nucleus. Now, as we're looking at the distinction between inside the nucleus and outside the nucleus, please keep in mind most of what we're going to be referring to, whether I write notes over here, is stuff that's happening inside the nucleus. So keep that in mind, regardless of what I'm writing over here. It's the only space I have to include information. All right, so when we look at our strand of DNA here, there are a few things that we do need to keep in mind. First off, I'm going to be using the word gene quite a bit when talking about DNA, and just know that gene is a section of DNA or a piece of DNA that is useful. So any section of DNA that is useful, any section of DNA that I'm referring to during this process known as transcription, I'll refer to as a gene, so keep that in mind as I'm going through this content. The other thing is, what's the main goal here? The overall goal, the overarching goal, if you will, of transcription is to be able to take the information in the DNA, within the gene, whatever section of DNA that we're looking at, and get that information to the ribosomes that are synthesizing proteins. Now, there's a second part to this, which is translation, and we'll cover that later, separate video. So for transcription, keep in mind the goal here is to be able to get the instructions from DNA out into the cytoplasm, because that's where the ribosomes are. We know that DNA has to stay inside the nucleus, one, because DNase will break it down, and two, because it's too big to actually pass through the nuclear membrane. So we have to find a way to get that information, which is stuck in the nucleus, out into the cytoplasm without getting DNA out there where it could be damaged, right? So that's the main goal here. So everything we're going to be talking about is how we get that information stored in the DNA, within the genes, out into the cytoplasm. All right, so keeping that in mind, looking at our diagram here, a few things to know about these labels. First off, we have these H's, which represent histone. Histone is a protein that allows us to wrap or spool DNA around it. You'll see it's represented in several parts on here. This allows us to take something that's actually a really big molecule, long chain, could be potentially inches long, and have it reside within the nucleus, which is microscopic. Keeping in mind, cells are microscopic, and this is an organelle inside of that cell. So histone allows us to wrap up DNA and kind of spool it in such a way that it will fit within the nucleus and hopefully prevent it from getting tangled up. Ideally, that is the main function. So histone's represented in there. In this state, obviously, we're going to be using DNA, using the genes, because, once again, they're useful to us. So it's not too tightly compact and wound up like we would see during that chromatin to chromosome shift during cell replication, or DNA replication and cell division, however you want to phrase that. All right, so that's our H's. From there, you're going to notice that we have an R. That R is going to represent our regulatory sequence. There we go. I'll also refer to it as a regulator as well. Essentially, one way to think about the regulatory sequence is to picture it kind of like an on-off switch for a gene. Keeping in mind, the entire strand of DNA is going to include lots of different genes, thousands of genes, up to potentially 20,000 genes within a single strand of DNA. And if we're trying to send instructions from the DNA out of the cytoplasm, we're going to be very specific and very intentional about what we're actually trying to do. So if we're only trying to focus on one specific gene, let's say, for example, a gene that allows us to make muscle cells, then we're going to want to ignore any genes that are not muscle cells, like, for example, this bone cell. Maybe over here we have skin cells. Maybe more further down here we have specific enzymes that we need to build for chemical reactions. We want to make sure that we are focusing only our attention on the gene that we need to work with, the one that we need instructions for. So the regulatory sequence, or the regulator, is going to identify this gene as one that is on and hopefully identify the rest that we don't need as off. Okay? So our regulatory sequence is providing us with this on-off switch for each of the genes. Now, it does this using other proteins. Proteins play a big part in what we're doing, and it's also the goal of what we're trying to accomplish here, protein synthesis, right? So for on, we're going to have things like activator proteins, that will bind to the DNA, and for off, we'll have things like inhibitor, also referred to as repressor proteins that will bind for the off position. Wait, I didn't mean to write bind, I meant to write proteins. I'm going to leave that mistake in there because, you know, I'm human. All right, more evidence that I'm human. Fantastic. All right, so for on, we're going to have these activator proteins that are basically funneling our enzymes and our chemicals into the nucleus and kind of herding them to where they need to go. Our inhibitor or repressor proteins are going to be blocking genes by literally blocking them from being able to access that gene's information. And once we start looking at how this actually works, it should hopefully make a little bit more sense. Right now we're just trying to identify some features within our diagram. All right, now before we get into the other two letters, let's talk about what's actually going to be happening here so it makes a little bit more sense. What are we turning on? What are we turning off? And from who or from what are we turning things on and off? So I'm going to erase this. Remember, you can always go back and pause information if you needed to write something down. That's the benefit of the video. And this chemical that we're referring to is an enzyme, very similar to all the other enzymes that we've talked about that catalyze reactions. And it's going to be known as RNA polymerase. RNA polymerase. Sometimes you'll see it just listed as RNAP. RNA plus the P from polymerase is RNAP. So RNA polymerase plays a big role in protein synthesis, and it's going to play the biggest role in what we're looking at here with transcription. Okay? So RNA polymerase is going to come into the nucleus because it can, right? It can actually pass through the nuclear membrane, and it's going to look for genes, not actually looking for genes, it has no consciousness, but it's going to be moving around in here, and they're going to be inhibitor proteins that are blocking certain genes. We're not trying to make bone cells here, maybe we're trying to make muscle cells, and so our activator proteins will be up here kind of directing RNA polymerase to that gene. We'll have inhibitors that are blocking RNA polymerase from actually having access to these genes, all right? So we want muscle cells, we don't want bone cells. So these inhibitor proteins, the on and off switch from the regulator, is going to direct these proteins to where they need to go. And so we'll have these inhibitors that show up and block access to bone genes, but keep the muscle gene open and available so that it can be activated. All right? So RNA polymerase is moving in, It's got several jobs. The first job is to find its way to we've got it labeled here as P, and that is going to be the promoter. All right, so P is our promoter, or our promoter sequence. All right, the promoter is, one way to look at this to help you remember it, it's the parking place for polymerase. Specifically RNA polymerase. Obviously, that's the one that we're talking about here. It's the parking place for polymerase. Before I can come into the building and do my job of teaching people, I have to go and park. So I got to know where to go. I'm going to park at the right building, the right entrance to get into the room that I need to be in so that I can actually do my job. So RNA polymerase is going to come into the nucleus. Here I have a gene that is on, based on the regulatory sequence. It says this one's on, and I've got my protons over there going, this is where you need to be, right? My activator protons are saying, come this way. My inhibitors are blocking this area off, so RNA polymerase is moving towards the muscle cell. It needs to know where to start. So it's going to park right here at the promoter. This is essentially starting point for the gene. If I need instructions on how to make something, it makes sense if I start at the beginning, or the starting point. So we start at the beginning of the gene, which is going to be identified by the promoter. If I need to know where to start, I also need to know where to stop, and that's where T comes in. And this is going to be my termination sequence. Which is also known as the terminator, which I think just sounds cooler anyway, so that's probably what I'm going to be calling it, the terminator, right? So the termination sequence, if the promoter is the parking place, it's where you go first, it's the starting point of the gene, if you will. Then the terminator, based on the word termination, means to end, this will be the end point of the gene. So RNA polymerase is going to come into the nucleus. It's going to go where there are proteins that are activating. They're saying, hey, this is where you need to be. The regulatory system has turned it on. This one is turned off. There's inhibitors that are blocking access to it. So RNA polymerase is not going to spend too much time here before it ends up where it needs to go. And when it gets to this muscle gene, it's going to go, this is where I start and this is where I end. We want to make sure that we know where we start, we know where we end, because the sequence is what's most important here. This order of all these nitrogen bases determine the sequence, which determine what we're going to be making or what we're going to be doing. So we got to know where to start, we got to know where to end. And that covers all the content that I have up here that's labeled with just letters or symbols so that you understand what we're going to be talking about here today. All right, so what's RNA polymerase actually going to do? This is the main point here of transcription. What is RNA polymerase actually doing to help us get the instructions from the nucleus in the DNA out into the cytoplasm where the ribosomes can do their job? All right, so here we go. Now that we know where to start, now that we know where we end, here's what RNA polymerase is going to do. Step one, it comes in the nucleus, it finds a promoter, and it begins to unzip the DNA. Now this should sound somewhat familiar when we looked at DNA replication. For DNA replication, when we make copies of DNA, we have to unzip or separate the two strands of DNA so that we know what to make copies of. In this case, we need to read the instructions that are encoded within that DNA strand, within that gene. So when we get to the promoter, we're going to unzip the DNA. RNA polymerase is then going to read the DNA. And it might look something like this. It comes over here, finds the promoter, knows where the termination sequence is, right? Because that's labeled for us as well. It takes that DNA and it unzips it so that we can read what those nitrogen bases are. And maybe we have an A and a C and a G and a T. Now, granted, we're going to have a lot more nitrogen base pairs here than just 4, but I wanted to make sure we included at least all 4 of the ones that are unique to DNA. All right, so we have our A, our C, our G, our T, our nitrogen bases. We've unzipped the DNA, we've read them, we've identified them, right? So then we're going to actually get to the transcription process. We're actually going to transcribe it. So what RNA polymerase is going to do is going to find the complementary base pairs for RNA, not DNA. So we're going to find the complementary base pairs for RNA. All right, so over here, we have A, C, G, and T. We need to find the complementary base pairs, very similar to how we do it with DNA. Anywhere there's an A, we're going to have a U, because RNA is a little bit different than DNA. Well, it's actually quite a bit different. But instead of thymine, RNA has uracil. So anywhere we see an A instead of a T, we're going to bond it with a U for uracil. Anywhere there's a C, we'll have a G. Anywhere there's a G, we'll have a C. And anywhere there's a T on the DNA, we're still going to have an A because RNA still has A's in it. All right? So the bases for RNA are going to be uracil, right? Uracil, guanine, cytosine, and adenine. So wherever we had thymine, then we now have uracil. So now we've identified the complementary base pairs for RNA, including the new one that we just introduced, which is uracil. From there, we're going to build RNA, but more specifically, mRNA. Now, anytime you see a little letter there in front, it's probably going to identify that RNA for having a specific purpose. And the nice thing about RNA is that we label it based on its job. So it should be pretty easy to figure out what its job is based on what that word is in front of RNA. In this case, mRNA, that M stands for messenger. So we're going to build our messenger RNA. Because we already know what we need to build it. We figured out our complementary base pairs. So from there, we build our mRNA. It'll be a single strand. It'll have our 4 bases. For example, we're simplifying this of U, G, C, and A. And this right here is our mRNA. Now, after we build the mRNA, down here, what happens to the DNA? The DNA just zips right back up. So it zips back together. I've been using the word re-zips. I don't think that's a real word. I just kind of like the sound of it. So it just kind of re-zips or zips back together. And what we had before looks like it did before we started messing with it. So we move back in here. And our DNA is all back together with its complementary strand. All right? Now, during this process, you'll notice that it only read one side. It only read one half of the DNA strand or of that gene. And that's because only one side has the information that we need to synthesize proteins. Okay? One half contains the instructions. The other half is only there to provide stability for the molecule. DNA molecules are very stable. They last a really long time, which is good because they need to last as long as you do to make sure they're still providing all the instructions that you need for growth, development, survival, reproduction. And obviously, through preservation techniques, it's able to outlast you as well, but we won't get into that. All right? So we read one half of it because only one half has the instructions. The other half's just there for stability. So after we're done with it, we've read it, we've figured out what we needed, we've made our mRNA, it just goes back together. All right? So now our mRNA, because it's single strand, because it's smaller, it's just a portion of the DNA, it's small enough to exit the nucleus. It's also not going to be affected by DNase because it's RNA, not DNA. And as we recall, hopefully we recall, enzymes are very specific when it comes to which molecules they'll actually interact with based on shape. This molecule is shaped a little bit different than DNA based on its simple sugar as well as the single strand. So DNase is not going to affect it. It's free to leave and go deliver the message because it's a messenger RNA, which that message is the instructions for how to create that specific protein that we need for muscles. So it'll leave the nucleus and head out into the cytoplasm where hopefully it runs into some ribosomes because ribosomes are responsible for protein synthesis. Keep in mind, what was our goal? Okay? Our goal was to be able to take the instructions in the DNA and get them out into the cytoplasm, which is where the ribosomes are. And to do that, we couldn't just rely on DNA by itself. RNA polymerase came in, unzipped the DNA, read it, found the complementary base pairs for RNA, built the mRNA, and put the DNA back together. At this point, my mRNA is able to leave the nucleus and go find some ribosomes to synthesize some proteins. What happens after that is a different process known as translation, which is obviously related to this, but we'll cover that in a later video. A few things to keep in mind as we're looking at how this works. Excuse me. Anytime we're working with a specific gene, keep in mind the level of organization that we're going to use for teaching purposes doesn't necessarily reflect what's real. I've simplified this strand of DNA quite a bit. It's a little bit more complex than this. We could have anywhere between, 5,000 to 20,000 different genes with numerous nitrogen bases in between for the code. So it's a lot more complicated than we're making it. Obviously, we're simplifying it. Also, everything that I need to build a muscle may not be contained in just one section of that strand. In fact, it definitely isn't. It's contained in many portions of the DNA strand, multiple genes spread out. All the muscle's not really going to be together. It's not as organized as we'd like it to be. So we may be turning on and off different sections of that strand to pull the information that we need to actually synthesize the protein correctly. And aside from that, to make sure that we get it to where it needs to go and that it can do its job. So it's definitely a little bit more complex than what we're trying to do here. Now, aside from that, there are a few things to keep in mind. As we're looking at this process and why it occurs the way it does, hopefully it makes logical sense that transcription would have to happen in the nucleus, that RNA polymerase is necessary, obviously, for this process because of DNA ACE and the size of DNA in general. But there are a few other things to keep in mind. One, the idea that maybe we could synthesize proteins in the nucleus wouldn't make a lot of sense. If we were able to get ribosomes in the nucleus, we could maybe just pull that information straight from the DNA and tell the ribosomes what to do. Essentially, theoretically, maybe that would be possible. However, the issue here is, remember, DNA is too big to pass through the nuclear membrane. If we did this with proteins and built proteins within the nucleus, protein molecules can be very large, in fact, too large to pass through that membrane as well. So we ended up with a lot of trapped proteins that weren't able to go do whatever it is that they were intended to do. Aside from that, even if we were able to let them out, it would create an opening that would be probably too big. Some of the DNA might escape, some of this DNA might end up in there, and then we create some problems, right? And we don't want problems. Now, aside from that, excuse me, knowing when to turn them on, when to turn them off, there are a lot of other processes involved here where you may have questions on how does it know when it needs to do this, how does it know when it needs to do that, how does it really find where it needs to go? It definitely is very complex. I would say some of those questions will definitely be answered in AP Bio. So if you are going to take AP Biology, look forward to that. If not, we're really going to try to keep it as simple as possible in this case. Now, a few things that I do want to make sure that you do consider here is make sure you understand the difference between replication and transcription. DNA replication is all about making a copy of DNA. Transcription is not just making a copy. It's being able to read what you have and then use it. The analogy that I've seen before is if I were to take a textbook over a subject that I wasn't familiar with, I could open up that textbook, even if it was in a different language, and copy down everything that I see on a separate sheet of paper and make a duplicate of the book. That would be replication. I've made a copy. Do I know what to do with that information? Have I really learned anything? Is it practically useful for me? No. Transcription would be actually taking that book, reading what it says, understanding it, and being able to do something with it. So there is a difference between the two. Even though you're going to hear terms that are familiar, like polymerase, DNA polymerase is an enzyme as well that helps put those copies that we make back together. Obviously, when we're looking at the sections of the genes and unzipping the gene and putting it back together, some of the similarities between the two start to show up, but the goal is very different. The enzymes involved are different. Okay? So please keep that in mind. If you have any questions or any concerns regarding how they're different, refer back to your notes on replication and look for all the things that are different, because there are plenty. Right? Aside from that, I think I've covered it all. If you have any questions, don't forget to reach out to me or check Google Classroom for the information that I've already provided for you. Thank you.